Okay, guys, I'm just going to take us through this section now of the bacteria chapter. We're looking here at what we call the growth curve of bacteria. What I want to do is I want to put this in context, and I want for you to imagine bacteria, we'll say, colonizing a new surface. Like, for example, bacteria have now colonized, we'll say, some sample of meat. It could be salmonella or it could be E. coli, have somehow gotten onto the meat. Or it could be another situation as well. For example, it could be streptococcus, which is a spherical type bacteria that invades your throat and colonizes your throat and obviously causes you harm. So, what we want to talk about today is the population growth curve. How does the population change over time? Now, of course, inside your body, you know that when bacteria enter the body, it's probably not just going to be one bacteria, it's probably going to be many bacteria. So the starting population mightn't necessarily just be one, but it could be maybe hundreds or maybe thousands that get into the body. But of course, once they find the surface that they are going to colonize, they will then take a while to get used to that surface, and then the reproduction will happen. Remember, the reproduction we're talking about here for bacteria is called binary fission, and binary fission is just where you start with one bacterial mother cell, and that mother cell will replicate its DNA. It'll elongate, it'll split after a cleavage furrow has formed, and it'll form two new daughter cells. So this is our starting point over here on the left, and what we're talking about here down the bottom is time. So time is passing here, and this could be a matter of minutes that we're talking about here, or it could be hours, depending. We're talking about the population of bacteria here on the left, and this is the population, obviously going up in increments there of 10,000s. Okay, so 10,000, 20,000, 30,000, 40,000, 50,000, etc. And this could be 50,000 bacteria per centimeter squared. Okay, so if we're looking at a surface. Right, so let's think about what happened now. I'm just going to take one example now, and I'm going to pretend we're talking about salmonella on meat. So that let's say now we have meat left out on the table, and a bacterial cell has fallen here onto the meat, and at this stage then we're undergoing the first stage of the bacterial growth curve, and this is called the lag stage. LAG. Now the lag phase is a representation of the time it takes for the bacteria population to kind of just get used to the environment. So, what do they have to do? The bacterial cells, when they come out of, we'll say, falling on the surface, many of them will probably have been in endospores. So, they'll have to exit the endospore. Remember, the endospore is the resting stage of the bacterial cell. Once they have emerged from the endospore, the next thing they're going to do then is they're going to try to choose the correct enzymes to break down the food. And once they have, I suppose, started metabolizing and growing themselves, then they start to reproduce. So once the lag phase is over, um, we now know that the population is ready to take over the surface of the. In this case, we're talking about salmonella taking over meat. So the conditions are ideal now at this moment in time. We're really ready for a population boom, and this is exactly what happens in the log phase. So after the lag phase, where there's kind of very, we'll say, a steady amount of bacteria, there's not really much growth. The log phase will happen next. So the log phase is basically a population boom, and the population boom will go up here as a steep increase here in the population of the bacteria. And only when it starts to level off again is it entering a different phase, i.e., the stationary phase. So, why is a log phase so successful for a bacteria population? Well, they have the ideal conditions, like, for example, the ideal amount of oxygen, the ideal amount of food, the ideal amount of space. There is very little toxicity in the environment. So the bacteria really have the ideal temperatures as well. So they really, and we're talking about bacteria in the log phase, everything is favoring bacterial growth. And then it reaches a point in time where conditions now are still maybe good, but maybe not as good as what they were down here. And the population at this stage is very large. And you're just talking about, again, if we're talking about salmonella, we'll say on a sample of meat. We could be talking here about the fact that there might be less room here for the bacteria to colonize. What we're starting to notice here actually is the environment's becoming a little bit toxic. Because there's so many bacteria, they're actually competing with each other. And that competition means that the bacteria are going to start fighting each other chemically. And how they start fighting each other chemically is they start to release something called antibiotics. Antibiotics, remember, are chemicals made by microorganisms that will kill or slow the growth of other microorganisms like bacteria. So at this moment in time, actually, at number 3, this is called a stationary phase. It's stationary because it's not really moving, and it's stationary as well because the birth rate is equal to the death rate. So actually, there's no increase in the population at this point. It's going to hit here about maybe 55,000, and it's not going to increase. And then there's a sudden change. You can see there's a sudden change here about 50 hours in here to this into this population curve. What we can see is there's a sharp decrease here in the population. Now, why would there be a sharp decrease here in the population of the bacteria? Well, the reason could be that the environment now is very toxic. Bacteria, what they tend to do is they tend to change the pH in the environment. So if if there's enough bacteria, what they do is they really make that environment quite toxic. And that toxicity means it's going to mass kill many of the bacteria. Conditions at this point really are very unfavorable for growth. It could be that the bacteria themselves will kind of generate their own heat. So it could be too warm, the temperature could be too high. There is just far, far too much competition. There could be too many antibiotics in the environment. There could be, as we said, there's very little space. They could be running out of food, and eventually just there's no food. So the bacteria's population sharply declines down here, and this is therefore called the death phase or the decline phase. So both are actually relevant here. Finally, then we enter what was called the survival stage, and it's very similar to the lag phase, I suppose, in that the population kind of remains relatively steady. Okay, there might be a little bit of a decrease, but not much. And in the survival phase, this is when the bacteria are starting to produce, trying to protect themselves, and they turn into endospores. So that process of endospore formation, remember, is when a bacteria will develop a thick outer coating around its DNA. And the reason it's developing a thick outer coating is to protect its DNA from being destroyed. It's very much like if you can imagine a seed. That's kind of what it is. It's like a seed that, when conditions turn favorable again, it can then come out of its seed. So endospore formation is happening here. The population remains steady. There is no real increase or decrease. It just remains steady. They're basically just surviving and putting up with the harsh conditions until conditions return favorable. And if conditions return favorable again, well, maybe this whole growth curve will happen again. So what we can say here for bacteria, and any bacteria, every bacteria population out there will go through these phases. And if we can recognize where the bacteria are on this growth curve, maybe we can manipulate it for our own purposes. So just to revise, the very start of this graph, when you see here about a steady amount of bacteria, but it's not really increasing very much, we call that the lag phase, LAG. When you see a sharp increase in the population, we call that a log phase. And it's a phase because it happens over a certain amount of time. If you see that there's a steady population after the log phase, in other words, it's leveling off, that's called the stationary phase. If you have a sharp decrease in the population, you call that the death phase or the decline phase. And finally, if you have a situation where the population is steadying off again after the decline phase, we call that the decline phase, or we call that the survival phase. So as I said, there are different reasons for all of this, and the best thing you could do really is you could read this in your own time to see does it make sense for you. Okay, but you're going to have to be able to read off these kind of questions here, and they come off quite often if bacteria are coming up. And the reason they come up is because we can use this for industry. Now, I will pop up a video on this in time on Google Classroom, but um, in industry, we're well aware of these growth curves. And if we can manipulate the growth curve, then maybe we can make sure that if we're using bacteria to produce, for example, milk or cheese or alcohol or different things inside, and we'll say industry, well, maybe we can make sure that we manipulate it so that they're producing the most amount of product as possible. So inside in the lab, or inside in the, we'll say the industrial plant, the food processing plant, you'll have people wearing white coats and they're working on their computers and they're trying to figure out how many bacteria are present in the milk or the cheese or whatever. And what they want to do, and if you think about this, the best phase you want to keep the bacteria in is the log phase. If they're in the log phase most of the time, then that means you can produce the most product possible. Okay. Now, in relation to industry, there's two different approaches that are taken by industry. So you can either have what we call batch flow, or you can have continuous flow. So what you need to know about this, and we're going to put it in context of, like, for example, the batch flow. For example, we can think about the dairy industry. So any cheeses that you eat will be produced as a result of batch flow. The word batch suggests that you're producing things in groups and then you're starting again. And that's exactly what this process involves. Batch flow processing involves a bioreactor. The bioreactor is a vessel that's used to produce a product by a microorganism. So this would be considered a bioreactor because it's just a piece of machinery that's used that bacteria live in, and they'll produce the product for us in here. So this could be, for example, cheese, or it could be alcohol, etc. Inside in this bioreactor, and by the way, we call it a bioreactor because it's a living environment. Inside in this bioreactor, we are maintaining the ideal conditions. So you have these guys here in lab coats checking the temperature, checking the pH, checking the pressure inside here, checking the numbers of bacteria as well. And they're constantly monitoring this environment to make sure that conditions are ideal. The next thing then is the bacteria will be, so if I start off here, the very start of this process for batch flow might mean that I put my milk in here, my milk to come from dairy cows, into the container. And I'm then going to add in, so these guys here might have added in the bacteria. So it would look like a powder when you add in this powder into your bacteria or into your milk. And the bacteria population then will start off, and we'll go back to that growth curve a second. They'll start off in this phase here, which is the lag phase. They've just been introduced to the milk. So in the batch flow processing, we want the bacteria to go through the lag phase, then the log phase, and we're going to try to keep them in stationary phase. Once they hit that stationary phase, they will have converted most of the milk to the cheese. Okay. So in other words, batch flow processes will go through lag, log, and stationary, and we do not allow it to go through the decline phase. Okay, so if it's starting to decline, the process is stopped, the product is removed, and we start again. So in a batch flow process, all of this cheese will have been produced after lag, log, and stationary, and then at the end of that process, we'll remove all of the product. None of the product is removed, we'll say, in the middle of this process. We all wait for the end. It's almost like, I suppose, baking a cake or whatever. You put in your ingredients at the start and you wait a certain amount of time, and then you remove your completed cake. Okay? So at the end of the process, then, if we have the product has been removed, we empty the bioreactor fully, we clean it down, we sterilize it, and then we can start the next batch again. Okay, so that would be batch flow, the batch flow process that is used widely, for example, for producing cheese. Okay, The next thing then is the continuous flow process. And this continuous flow process, I suppose, we see it in more complicated setups, like, for example, in the pharmaceutical industry, if we're trying to produce a certain drug or chemical. So again, you have to use your bioreactor. And remember, a bioreactor is just the vessel in which the bacteria produce the product for us. And inside here, it could be some, I don't know, it could be some chemical like penicillin or something that we are producing for a pharmacy. So what happens? All of the nutrients are just like the previous process, all of the nutrients are added at the start. The bacteria are introduced at the start. But then the conditions are kept ideal again. So we have to look at the temperature, the pH, we have to look at the pressure, we have to look at the concentration of nutrients that's being added as well. We will make sure that all of these conditions are maintained and that they are the ideal conditions. But one difference between the continuous flow and the batch flow is that instead of, I suppose, stopping this process and starting again, we are going to try to keep this in continuous flow. We're going to try to keep it going for a long period of time. So if I was in the milk industry, we would have cheese produced maybe after a day or 2 days. But for continuous flow, it might mean that your process is working over 6 months. And you have scientists every single day whose job is to make sure that this process continues. So how do you make sure this process continues? What you do is you continually add nutrients. So every day you're adding in nutrients here so that your bacteria have nutrients to survive. Every day as well, you are removing product. So we're removing product here from the bioreactor. So we're trying to drain off some of the penicillin, for example, that we have been producing. So they are the two big differences between the batch flow and the continuous flow. Batch flow goes through lag, log, stationary, and once the stationary phase is hit, we remove all of the product. Continuous flow is where we go through lag and log, and we try to keep it in the log phase. We do that by careful monitoring. We continually add nutrients and we continually remove product. Unlike the batch flow, batch flow, remember, all the product was removed at the very end of the process. Nothing is removed in the middle. Whereas continuous flow, we are continually removing the product from this. Okay, so in theory, the continuous flow could be maintained forever, but it's very difficult to maintain the continuous flow process for we'll say, the chemical industry. So why is batch flow generally preferable? Because of this, because it's very difficult to maintain the optimum conditions. It requires a very good scientist who's studying everything very carefully. And also, I suppose another reason is because in relation to batch flow, like it's everything can be sterilized every single time once you've completed your batch and you start the next batch again, so things don't become too toxic, whereas that issue can arise in a continuous flow process. Okay, so that is how industry use this, and again, there's notes on this that you can study to get a little bit more detail on it.